Hello, dear students. I hope you all are fine and doing well. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, we are in a continuation of the chapter that is Principles of Inheritance and Variation. In previous lecture, we have already understood that what are the different terms which are going to be used in this chapter and what are the technique used by Mendel for the studying this genetics on inheritance patterns. In today's lecture, we are going to start with the experiments which was performed by Mendel. And in that, we are going to understand first the inheritance of one gene. Means how one gene is getting inherited from parent to the another parent to the offspring without considering the other gene. Okay, so we are going to talk about inheritance of one gene which is responsible for one character. Okay, so monohybrid cross we are going to discuss. We know that for the crossing a pea plant, we already understood in the previous lecture that how the crossing performed by Mendel in a pea plant. We know that there is a parental generation. Parents are there, and parents have a bisexual flower with the carpel and stamen. And we already understood that the stamen are removed from the one particular desired plant, and pollen grain are inserted over the carpel, stigma of a carpel, to carry out a fertilization event. So, inheritance of a single gene is obviously known as a monohybrid cross. So let's talk about this monohybrid cross, which deals with the inheritance of a single gene or single gene coding for a one character. So what is mean by monohybrid cross? When a cross is made between two parents differ in a single character, means two plants we are going to take in or two parents we are going to take in, and both plant parent have a alternative forms of this character that is traced. Means one trait is present in one parent and another trait is present in another parent. Okay, so when a cross made between like such type of parents, then it is known as monohybrid cross. Now, Mendel crossed tall and dwarf pea plant to study the inheritance of one gene. And we know that tall, one parent is tall and one parent is dwarf. Okay, so this tallness and dwarfness are the traits of a character, that is height. What he did, he collected the seeds produced as a result of cross and grew them to generate a plant of the first hybrid generation. Means what? He taken a tall plant, dwarf plant, made a cross, and we produced the hybrid plant and grown hybrid this plant and collected their seeds of this first hybrid generation. This generation is also called the first filial progeny or the F1 generation or first daughter generation. Filial means daughter. First daughter generation, first daughter progeny of the first generation. Okay, and carried out cross like this. So what he did during the cross? See, here you can understand properly. This is the P generation, means parental generation. Okay, this is a parental generation, and here parent is one is homozygous tall, and another one is the dwarf plant. Tall plant is homozygous tall, dwarf is homozygous dwarf, that is a small t, it is capital T capital. These both individuals are diploid individuals, so we are writing capital T and small t small t. But when the offsprings are produced or gametes are produced, the gametes will carry only one allele out of these two. So capital T will be there, and here small t will be inherited in the gamete. After this, here gametic fusion occurs. Means gamete are going to fuse. When gamete fuse, they form a heterozygous plant, that is capital T small t in a F1 generation. When the F1 just see all whatever the tall, if you take homozygous and the recessive you dwarf plant you will take both homozygous, then obviously the F1 generation will be capital T small t due to the gametic fusion. And this F1 is capital T small t, its phenotype is going to be tall because dominant trait is tallness is the dominant trait. So in heterozygous condition, tallness will be expressed. After this, the F1 we carry out a self-fertilization. What we carry out after F1 generation? We carry out selfing or self-hybridization. And this self-hybridization is going to produce the F2 generation. In a selfing, what we are going to do? We are going to cross F1 capital T small t with the F1 capital T small t. Now this I am taking a male and this I am taking a female. So male have two types of gamete, that is capital T and small t. Okay. And this capital T small t having also two type of gamete, that is capital T and small t. This is we are considering female and this we are considering as a male hybrid. Now when selfing is done, the F1 gametes are like this and we make a cross, then F2 generation is obtained. In a cross, we will find that when capital T gamete cross with the capital T gamete, it is going to produce capital T plant. Similarly, when capital T cross with the fuse with the small t gamete, then it will produce a capital T small t. Here also, when small t fuse with the capital T small t, it is going to produce a capital T small t plant. And here, when small t combine, they is going to produce a small t plant. Here you can say this having a tall phenotype. This is having a tall phenotype. This is also having a tall phenotype. Here also you can see tall phenotype, but here you will see the dwarf phenotype. So out of total 4 offspring in the F2 generation, 3 are tall and 1 is a dwarf or short. So phenotypic ratio, if we see, then it is a 3:1. Okay, phenotypic ratio is 3:1. But if we write a genotypic ratio, then the 3 genotypes are seen here. Capital T is to capital T small t is to small t small t. Capital T is 1, capital T small t is 2, and small t is 1. So genotypic ratio of this monohybrid cross is 1:2:1, whereas the phenotypic ratio is 3:1. Okay, so this is for the one character inheritance or monohybrid cross. Okay, dear students, I hope you understood this. Now, we have to understand this in more detail. So let's talk about some of the statements. So what he did in this experiment, as we had discussed, he made similar observations for the other pairs of trait. Means we have done a cross with the only one character, that is the height. But Mendel used 7 characters. For such 7 characters, 7 different crosses were made. So he made similar observation for the other traits, other pairs of traits, means other 6 characters, and found that F1 always resemble the dominant parent. Yes, students, this is the one of the important point that you have to remember. F1 always resemble the dominant parent. Means whatever the phenotype is expressed in the F1, that is going to be dominant trait. Yes or no? That is going to be dominant trait. So F1 have a dominant trait. Now, Mendel observed that all the F1 progeny plants are tall, like one of its parent. None were dwarf, means tallness is a dominant trait. And that the trait of the other parent was not seen in them, means dwarfness is not seen in them. So Mendel then self-pollinated the tall F1 plant. What did he do? Self-pollinated, self-fertilization of F1 is done. Means F1 tall plant, that is capital T small t crossed with the capital T small t. In the second filial generation or F2 generation, some of the offspring produced were dwarf, and that are the product of what? Self-fertilization over the F1 plant. So some offspring were produced were dwarf, and we have studied that dwarf is only one. This dwarf character was not seen in the F1 generation but expressed in the F2 generation. Means you can understand that this dwarfness is present in parent, absent in F1 generation, but again get expressed in the F2 generation. The proportion or probability of the plant ovulating F2 generation is 1/4 dwarf and 3/4 tall, because 1 is dwarf and 3 are tall. So the tall and dwarf traits were identical to their parental type and did not show any blending. Dear students, please remember this important point. Here there is no mixing of character. Blending means mixing of character. You will not find any mixed character, intermediate character here. You are going to see only parental traits in the offspring. So tall and dwarf traits of the offspring are identical to their parental, and any intermediate or blended expression is not observed. Any mixed expression is not observed. They're like semi-tall. You will not see like that. Okay. That means all the offspring were either tall or dwarf. None were of in between height when monohybrid crosses follow. Okay. Will not find any plant of intermediate height. Next, only one of the parental trait was expressed in F1 generation, while the F2 stage show traits were expressed in a proportion of 3:1, means phenotypic ratio is 3:1. So what would be the phenotype of a plant that had a genotype capital T small t? So capital T small t is going to be tall plant because capital T is dominant over the small t, or we can say tallness is a dominant over the dwarfness, and the only dominant phenotype is going to express in the F1 generation. So F1 plant capital T small t is going to be tall plant. Okay, so this is the thing. Now, if we talk about capital T, a pair of allele, and a small t as a pair of allele are called a genotype of the plant. While the descriptive terms for phenotypic, which are expressed morphologically, okay, that are tall and dwarf, are the phenotypes. Mendel found the phenotype of the F1 heterozygote exactly like the capital T parent in appearance. So he proposed that what he seen Mendel, what Mendel did, Mendel seen a capital T small t plant, F1 plant, and capital T plant, parent plant. So he observed the similar expression. Both are of approximately similar size. So he proposed that in a pair of dissimilar factors, one dominates the other. One of the important things. What he proposed that in a pair of dissimilar factor like capital T and small t, one dominates the other, as in the F1, capital T is dominating over the small t. Hence is called the dominant factor, okay, that is capital T, and the recessive factor is the small t. And similarly, we can assign it to the other character as well. Okay. So if one character is not expressed in F1 generation, then what is called as recessive? That which the whatever the character trait which is not expressed in the F1, it is recessive, and which get expressed, it is a dominant. So we have already understood this tallness and darkness. Other characters are also here like this. So other characters are like this: seed shape, round, wrinkled, seed color, yellow, green, flower color, violet, white. We have discussed already this in the medals technique also. What character we experimented. Other are port shape, which can be full or inflated or can constricted, which is a recessive trait. Then green, yellow, port color, dominant and recessive trait respectively. Same way flower position can be axial or terminal. Okay, these are the other 6 characters. But in the first character, as you have seen, capital T is the tallness and it's the dominant over the dwarfness. Dwarfness, small t is the recessive. Now, alleles can be similar as in case of homozygotes, that is capital T or small t, or can be dissimilar as in case of heterozygotes, that is capital T small t. As genes are controlling one character height, capital T small t heterozygotes is a monohybrid. Means capital T small t is an obvious hybrid plant of the parental tall and dwarf, but it is hybrid for the single character, single trait, so we call it as a monohybrid. The cross between capital T and small t is a monohybrid cross. He observed the recessive parental trait is expressed without any blending in the F2 generation, as we have discussed. Tall and dwarf plants produce gametes by the process of meiosis. The alleles of the parental pairs separate or segregate from each other, and only one allele is transmitted to a gamete. Dear students, one of the important points that you should mark here, that in a parent, the alleles are going to be occur in a pair, but they are going to separate, and only one allele is going to transmit it in gamete. See, suppose parent is capital T means in gamete only capital T will be obtained. If parent is small t, and then in gamete we will obtain only small t. But if parent is capital T and small t, then two types of gametes are going to form. One gamete having the capital T and one gamete having the small t. But gamete will have only one allele at a time. Two alleles cannot be present of the same character. So segregation of allele is a random process. It is a random process. There is a 50% chance of gamete containing either allele. If heterozygous condition is there, then 50% gamete is going to have a capital T and 50% gamete is going to have a small t. And in one gamete also, the chances of it becoming a capital T or small t is a 50%. The gametes of tall capital T plant have the allele capital T, and the gamete of the dwarf having the capital small t allele. During fertilization, the two alleles capital T from one parent through the pollen and small t from other parent through the egg are united to produce the zygote, and they produce a zygote. Now this zygote that has one capital T allele and one small t allele, that hybrid have capital T small t. Since this hybrid means hybrid plant which is going to develop from the capital T small t zygote. Okay. Now this hybrid contain alleles which express contrasting trait. The plants are heterozygous. Punnett square board is commonly known as, in the first cross which we understood, the Punnett square is used. And this Punnett square board is commonly known as checkerboard. The types of gamete produced by the parents, the formation of the zygote and the progeny can be represented in a diagram, in a one diagram, that is Punnett square board or checkerboard. It was developed by British geneticist. This Punnett square is developed by British geneticist, and his name is Reginald C. Punnett. And because of this, we called it Punnett square. It is a graphical representation to indicate all possible unions of gametes during fertilization. And this is the Punnett square matrix, which is developed by Reginald C. Punnett. See, this is the Punnett square. In Punnett square, okay, here we will going to put these gametes. These are the gametes. Here you will put male gamete in this side and female gamete in this side. Okay, this is the male gamete capital T small t, female gamete capital T small t, and we make a fusion. This fuse with this one, then this gamete is going to fuse with small t, then this small t fuse with this gamete, and this small t again fuse with this small t gamete. And like this, genotype is going to be observed by the fusion of gametes, and these are the phenotypes which are going to be observed here. Okay, so this is a Punnett square. So phenotypic ratio is 3:1. Okay, and the genotypic ratio is 1:2:1. So probability in relation to genetics. Okay, in genetics, what is the probability is used here? So how the probability is used? So Mendel's law of segregation and independent assortment reflect the same laws of probability. In Mendel's laws, we will see the function of this probability. And in this, you will see that tossing coin or rolling dice having a probability. The probability scale range from 0, an event with no chance of occurring, to 1, an event that is certain to character. Means in a capital T, it is fixed that probability of getting capital T is 100%. So we can write 1. In capital T parent, small capital T gamete is going to be produced. That is 100%. So the probability is 1. But if we talk about the probability of small t from the capital T parent, then it is going to be 0. Okay, so it can range from 0 to 1. For example, the probability of tossing a head with a normal coin is half. Yes, now when we toss a coin, the chances of being a head or a tail is 50%. Okay. Tossing a coin, probability of falling heads is 0.5%, 50% or half. And from probability of falling the tail is also 0.5. Okay, because it either can be head or tail. Okay. Now, so the chances are 50%. Okay. Same way for a dice, The six possibility are there. Either it can be one, two, three, four, five, six. So probability of one is one because six faces. Dice have six faces. So at any one face, it is one dot is given. So chances of one is one by six. Chances of two dot on a one face is also one by six. And same way for three, four, five, or six dots on a one side of getting a that side is one by six. So probability of falling a one or two or up to a six is one by six. Like coin toss, each ovum from a heterozygous parent has a half chance of carrying a dominant allele and half chance of carrying a recessive allele. Okay, so it can be 50% capital T or it can be small t. And to calculate the probability of all possible genotype of offspring in a genetic cross, the possible gametes are written on two sides, usually the top row and the left column. All possible combination of the gamete during a fertilization are represented in boxes below the squares. Okay, so in boxes you will see the possible combination or fusion. The parental tall and dwarf small t plants, the gametes are produced by them and the F1 progeny is capital T small t. The F1 plant of genotype capital T small t are self-pollinated. The symbol differently used for the male and female, then you can see this symbol is used to denote the female or egg, and this symbol is used to denote the what male or pollen gamete of the F1 generation. The F1 plant of the genotype capital T small t produces a gamete of genotype capital T and small t, means F1 is going to produce two gametes, that is capital T and small t in equal proportion, that is 50%. When fertilization takes place, the pollen grain of genotype capital T have a 50% chance of pollinate egg of the genotype capital T as well to the genotype small t. If pollen grain has a capital T, either it can fuse with the capital T or a small t, 50% chance. Also, the pollen grain of genotype small t have a 50% chance of pollinating eggs of genotype capital T and as well as genotype small t. As a result of random fertilization, the resultant zygote can be of capital T, capital T, capital T, small t, or small t, small t. And from the Punnett square, it is easily seen that 1/4 probability of 0.25 of the random fertilization leads to the capital T, then one half probability, that is 0.5, leads to capital T small t, and 1/4 for the small t small t. So though the F1 has a genotype of capital T small t, the phenotype seen is tall, that is in heterozygous condition. And at F2, 3/4 plant are tall, whereas some of them are capital T, and while others are capital T small t. Externally, it is not possible to distinguish between the plants with the genotype capital T, and capital T small t. Externally, we cannot differentiate. By seeing a tall plant, you can't say that it is a homozygous tall or heterozygous tall. Hence, in the two genotype capital T, and capital T small t, only tall is expressed and small t is not getting expressed. Hence, the character tall or allele capital T is said to be dominant over the other allele small t or a dwarf character. And due to this dominance of one character over the other, that is that all the F1 are tall, that is capital T small t genotype. In the F2, 3/4 of the plant are tall, though genotypically half are capital T small t and only 1/4 are capital T capital T. Out of 3/4 means you can see 3 are tall. Out of this, capital tall, one half are capital T small t and 1/4 are capital T due to the 1:2:1 ratio. This led to the phenotype 3:1, but the genotype ratio 1:2:1 that we have understood already. 1/4, one half, 1/4 ratio of capital T, small t, small t, small t. So read this important statement, second one. In this 1:2:1 ratio or capital T is to capital T, small t, or is to small t ratio is expressed mathematically condensable in the form of binomial expression. It can be expressed in a binomial expression that ax plus by square, that the gametes bearing capital T and small t in equal frequency of half. So the expression is expanded as given below. Half t plus half t whole square is equal to half t plus half t into half t plus half t is equal to half capital T, 1/4 capital T, 1/2 capital T small t and 1/4 small t. Okay. Now, Mendel self-pollinated the F2 plant and found that the dwarf F2 plant continued to generate dwarf plant in F3 and F4 generation. So we have just understood the F1 and F2 generation. If the fertilization occur in F2 generation, how they are going to produce F3 generation, and from F3 how they are going to produce the F4 generation. Then that we have to understand clearly. And to understand that, we will make a cross. But he concluded that the genotype of dwarf are homozygous multi. Okay. Now, if you want to conclude this, that how? So see, in parental generation, we know that there is a capital T, and crossing with the small t. So in F1, we know that what capital T, small t is obtained. Now, if we carry out a selfing, F2 generation is going to be produced. And in F2, there will be a capital T, capital T, small t, and small t, small t. Now, again, selfing of all these three is going to occur. And if selfing occur, all are going to be tall, capital T, capital T. If here selfing occurs, then that is going to produce three types of plant: capital T, capital T, small t, and small t, small t. This also undergo selfing, and if it undergo selfing, then it is going to produce a small t. So this is the F3 generation. So you can see in F3 generation, the selfing occur and like this offspring are produced. Now see, in a parent, the two types of offspring are there, capital T and capital small t. See, if we see now in F1, the capital T small t is produced. So capital T small t is 100%. All plants are going to be this one. But when we perform a selfing in F2 generation, out of this 100%, this 25% are producing capital T small t, 50% are of this type, and 25% are of this type. When selfing of this capital T is done, in this, all 25% offspring are going to be continued. Out of this 25, all 25% is going to be produce capital T. And this 25% dwarf is going to produce a 25% dwarf. But here, when we talk selfing of capital T small t, 50%, then out of 50%, 12.5% are going to be this one. 12.5% are going to be this one, and 25% are capital T small t. Now, if we perform selfing for the F4 generation, then this is continued to selfing to produce a capital T, that is 25%. Okay. Now, here this again undergo a selfing to produce a capital T, 12.5%, but it undergoes selfing. And then it produces capital T, capital T, small t, small t, small t. This is 25%. So 12.5% is going to be this one. 6.25% is this one. 6.25% is this one. This undergoes selfing and produce small t, that is 12.5%, and this also undergo selfing to produce 25%. Okay, such way this F4 progenies are obtained. Okay. Now, if we write in a corresponding generation about the number of homozygous and heterozygous in F1, F2, F3, and F4, if we write a corresponding of what? Homozygous and heterozygous percentage of offspring, then we come to know that in parental generation, 100% are homozygous and 0% are heterozygous. But in F1, it is completely opposite, 100% heterozygous and 0% homozygous. But in F3, we carry out a selfing of this heterozygous. Then in F2, remain with the 50% heterozygous and 50% homozygous, that is capital T and small t combined. Then in F3, you will see the heterozygous are 25%. Only heterozygous is this one. And remaining are this all are homozygous, means 75% is homozygous. And in F4, you will see the only 12.5% are heterozygous. The remaining 87.5% are homozygous. Means from F1 onwards, if we keep on continuing selfing from generation to generation to produce F3 and F4, then you will see that homozygosity is getting increases from generation to generation, whereas this particularly it is increases. Okay, whereas heterozygosity is decreasing from generation to generation. Okay, so that is the thing which is showing exactly what happens now and how the homozygous or pure lines are produced. Okay, I hope you understood this in detail. Then from the studying this monohybrid cross on the tall and dwarf plants and getting a ratio, the some laws was defined and two laws was assigned based on this monohybrid cross, that is the law of dominance and law of segregation. These two law was developed after this particular monohybrid cross. First is law of dominance. Law of dominance, what it explains? It explains that characters controlled by a discrete units called factor, means characters are going to be controlled by factors or a genes. And factors or a genes occur in pair. That is also known. In a dissimilar pair of factor, when there is a presence of dissimilar pair, if there is a similar pair, there is no issue. But if there is a dissimilar pair of factors pertaining to a character, one member of the pair dominates out of two alleles, one pair of two alleles, one dominates the other. Which one dominates, that is dominant allele, and the which get particularly not expressing, that is the recessive. And the law of dominance is used to explain the expression of only one of the parental character in a monohybrid cross. One of the parental. Monohybrid cross means capital T, tall is expressed in F1. So one of the parental character in a monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained in the F2. Okay. So this law of dominance explains us that capital T is dominant over the small t in F1 generation. Similarly, tallness is dominant over dwarfness. So when there is a dissimilar parallel allele, capital T, small t, capital T dominates the small t, and expression of capital T only is going to occur. Small t is not going to be expressed. So tall phenotype is obtained. And that is the law of dominance. But there are some exception also to the law of dominance. It is not universally applicable. Be remember, law of dominance is not universally applicable because there are exceptions. An exception which occur to the law of dominance are: number one, that is incomplete dominance, and second, codominance. These are exception to the law of dominance or law of complete dominance. Okay, and we will discuss this incomplete dominance and codominance after completion of the monohybrid cross in the later part. Another law is the law of segregation or law of purity of gamete. What is homozygous parent? We discussed already. Homozygous parent produce all gametes that are similar. So Mendel's law of segregation states that the two alleles of a gene, when present together in a heterozygous state, do not fuse or blend in any way, but remain distinct in a heterozygous condition and segregated during meiosis or in the formation of gamete. So each meiotic product or gamete will carry only one of them. Okay. So in a parent, they can be in pair. And in pair also, they are distinct. They are not going to fuse, or then they are not going to blend, or they are not going to undergo crossing, or they are going to mix. So after meiosis, they are going to remain as it is and obtained in a gamete. Okay. So gamete will carry, due to this meiosis, the gamete will carry only one of them. Though the parent contains two alleles during a gamete formation, the factor or allele of a pair segregate from each other such that the gamete receive only one of the two factors or two genes. A homozygous parent produces all gametes that are similar. A heterozygous parent produces two kinds of gametes in an equal proportion, each having one allele. Means capital T small t is there, then there is a chances of a gamete with the capital T and of small t as well in a 50% proportion each. Segregation of genes is a universal phenomenon, and in all organisms reproducing by a normal sexual method, it is a universal phenomenon. Means it is universally applicable. Okay, so these are showing a monohybrid cross. You can see here they have given another example of the red sweet pea and the pure white sweet pea. Capital R here, small r here. Here two gametes are produced, capital R and small r. So they produce only all particular offspring in F1 are capital R, small r, and that is the first generation, and that are of red color. But in F2, the red capital R, small r is going to fuse with the capital R, small r, and capital R, small r gamete are produced. Capital R, small r gamete are produced. In second generation, we find a capital R, small r into and small r, small r. So here also genotypic ratio you can see is 1:2:1, and phenotypic ratio is 3:1. Means this ratio shows that it is following a monohybrid cross, and law of segregation is occurring, where here you can see meiosis is occurring and gametes are produced. Here also meiosis is occurring, gametes are produced like this. Here it is a dissimilar pair, so 50% chance of capital R and small r. Here similar pair, so all are of same kind. So let's talk about some of the MCQ. Factors occur in a pair. Okay, you know that. Law of segregation explains phenotype in F1, genotype in F1, phenotype at F2, genotype at F3. So phenotype at F2. Okay. Law of segregation explains phenotype because the 3:1 ratio is obtained and two types of gametes are formed. The gametes produced by a homozygous parent. So if a homozygous parent produces gametes, they all are going to be similar. What is monohybrid cross? A cross between two parent differing in two characters, self-pollination, cross between two parent, cross between two parent differing in a single character. So it is a cross between two parent which differ in a single character, means which have a traits of one character. First generation of monohybrid cross is F1. Mendel self-pollinated the tall F1 plant to form F2. What are the units of inheritance? Units of inheritance is nothing but a genus. An alternative forms of genes is called alleles. Okay. So this is about the monohybrid cross and their laws which are derived on this. Okay. I hope you understood this monohybrid cross. Further discussion we will continue in the next class. Till that, thank you.